In this activity, you'll examine the structure of two different seeds, a garden bean and a corn kernel. The bean represents a Eudicot seed, while the corn kernel represents a Monocot seed. By examining both, you'll be able to compare how nutrients are stored, how the embryo is organized, and how Monocot and Eudicot seeds differ in structure. As you work through the activity, pay attention to how these structural differences reflect different strategies for supporting early plant growth. Seed formation begins with a process called double fertilization, which is unique to angiosperms. Two sperm cells enter the ovule. One sperm fertilizes the egg, forming a zygote, which develops into the embryo. The second sperm fuses with two nuclei in the central cell to form the triploid endosperm, a nutrient-rich tissue that supports embryo development. In some plants, the endosperm remains the main storage tissue. In others, those nutrients are absorbed into the cotyledons. One of the most important differences among flowering plants is the number of cotyledons, or seed leaves, in the embryo. In Eudicots, embryos have two cotyledons. These cotyledons often enlarge and store nutrients that the developing seedling will use during germination. In monocots, the embryo has one cotyledon, and the endosperm usually remains as the main nutrient storage tissue. The garden bean you'll examine today is a Eudicot seed, so most of the seed will be made up of the two cotyledons. Before we open the seed, let's look at the external structures you'll be able to observe right away. The outer covering of the seed is the seed coat, also called the testa. This layer protects the embryo from physical damage and drying out. If you look closely at the seed's surface, you should also be able to locate the hilum. The hilum is the small scar that marks the point where the seed was attached to the ovary wall inside the fruit. Near the hilum, you'll usually see a tiny opening called the micropyle. This opening was originally part of the ovule and allowed the pollen tube to enter during fertilization. As you examine your bean seed, try to locate each of these structures before removing the seed coat. These are the main structures you'll be looking for inside the seed. At the center of the seed is the embryo, which is the young plant in an early developmental stage. The cotyledons, sometimes called seed leaves, store nutrients that support early growth after germination. The plumule is the embryonic shoot that will eventually develop into the stem and the first true leaves. The hypocotyl is the portion of embryonic stem located below the cotyledons, while the epicotyl is the portion above the cotyledons, although the epicotyl may not always be obvious until the seed begins to germinate. Finally, the radicle is the embryonic root and is typically the first structure to emerge during germination. As you dissect the bean seed in the next step, use this diagram to help you identify each of these structures. Before we begin the observations, think about this question. Why might Eudicots store most nutrients in the cotyledons, while monocots store most nutrients in the endosperm? Consider how these different strategies might affect how seedlings germinate and grow. As you examine the bean seed, pay attention to where most of the seed's mass is located. Begin by examining the bean seed. First, observe the external structures. The outer protective covering is the seed coat, also called the testa. Look closely to locate the hilum, which is the scar where the seed was attached to the ovary wall inside the fruit. Near the hilum, you should see a small opening called the micropyle, which was part of the original ovule. Next, look at the right picture where the seed coat has been removed and the seed separated into its two halves. These are the cotyledons, which store nutrients for the embryo. Look for the small embryo attached to one cotyledon and identify the plumule, hypocotyl, and radicle. In your lab notebook, draw and label both the external structures of the seed and the embryo structures inside. Now examine the corn kernel, which represents a monocot seed. The thin outer layer is the pericarp, which is actually the fruit wall. At one end of the kernel, you may see a fruit scar where the kernel was attached to the cob. On one side of the kernel, locate the white oblong area, which marks the embryo. You can see it better from the outside of this dried kernel. Most of this region is the single cotyledon. Inside this embryo region, you can see the embryo, while the majority of the kernel is filled with yellow endosperm, which stores nutrients for the developing plant. First, draw a side view of the kernel showing the pericarp and the fruit scar. Then make a longitudinal section drawing, labeling the endosperm and embryo. Now that you've examined both seeds, take some time to compare what you've observed. First, compare the internal structure of the bean seed and the corn kernel. Think about where most of the stored nutrients are located in each seed. In the bean seed, most of the interior is made up of cotyledons, while in the corn kernel, most of the seed is filled with endosperm. Consider how this reflects the difference between Eudicot and monocot seeds. Next, compare the hypocotyl and the radicle. The radicle is the embryonic root, which is usually the first structure to emerge during germination. The hypocotyl is part of the embryonic stem, connecting the cotyledons to the root. Think about where each structure sits in the embryo and what role it plays when the seed first begins to grow. Finally, explain the difference between the plumule and the cotyledon. The plumule contains the embryonic shoot and young leaves that will eventually develop into the above-ground portion of the plant. Cotyledons, on the other hand, mainly serve as nutrient storage structures in many seeds. Use what you observed in your dissections to support your answers. Let's summarize the major differences you observed between the bean seed and the corn kernel. The bean seed represents a eudicot. It contains 2 cotyledons, and these cotyledons store most of the nutrients that support the developing embryo. Because of this, the embryo structures take up most of the seed. The corn kernel represents a monocot seed. It has only 1 cotyledon, and most of the seed is filled with endosperm, which stores nutrients for the embryo. You may also notice that the outer layer of the corn kernel is actually part of the fruit wall, called the pericarp, which is fused with the seed coat. These structural differences reflect 2 different strategies for storing nutrients and supporting early plant growth.